voluntarily ever. And you can only control skeletal muscle voluntarily sometimes. So for instance, can you, although my diaphragm is skeletal muscle and it makes me inhale and relaxes for exhalation, and I can control it voluntarily, kind of, because I can go, right? I can do that. Can I hold my breath to death? No, because it will let you control it voluntarily as long as you are a team player. When you do it improperly, it will take the damn keys away from you and tell you to sit your ass down. Okay? So my 18-year-old, she's my 18-year-old daughter, when she was like 4, we went to Toys R Us, and she wanted some $400 footstool toy. It was like a pony that was this big. It wagged its tail and it wiggled its nose, and its name was Buttercup. I still remember. And Savannah told me that if I didn't get it for her, she was going to hold her breath to death. And I was like, let's try. And so she did. She tried, and it didn't work. She didn't pass out, don't worry. But I did not only not get her the pony, but then I taught her why it didn't work. And I was like, your brain is smarter than you are, apparently, right now. So yeah, it's only sometimes voluntary, and the other two are never voluntary. And then locations in the body. This is called skeletal muscle because it's mostly attached to your bones, only mostly though. Like this one's attached to my bones, but my facial muscles, they're not moving bones, they're moving skin. So it's mostly attached to your bones. And then you find smooth muscle in all hollow tubes and organs that need to move things except the heart, right? That's got its own specialized cardiac muscle. But any place else in the body that you have a tube or a hollow organ that you need to move things through, there's going to be smooth muscle in the wall. Blood vessels, ureter, urethra, urinary bladder, digestive tract, all of the bronchioles in your bronchi and bronchioles in your lungs, all of those have smooth muscle in the walls. Go ahead.
Are they also the papilla or the, what's the name, the skin ones?
Oh yeah, those two. Yeah, a couple of other places. They also control the diameter of your pupil, and do a couple of, and the roundness of your eyeball. So any place you need to wiggle something that is not voluntary and not heart is smooth muscle. Yeah. So and then of course you only find cardiac muscle in the wall of the heart. The other name for the heart muscle tissue is myo, which means muscle, and cardium, which implies heart, so heart muscle, okay? Now let's talk about the speed of contraction and the ability to remain contracted. This becomes important in physiology. So one of these three tissues is never in any particular hurry, kind of like the sloth of the tissue world, but it goes forever. And one of these tissues is your emergency response tissue that'll get you out of the way of a bus or help you get away from a saber-toothed tiger, right? Which one is your emergency response tissue? Skeletal muscle, yeah. So that needs to contract the fastest upon stimulation, but because it contracts so quickly, it runs out of energy very quickly. So it's very difficult to maintain a contraction. So the metaphor that I would like to use for these is sprinter, jogger, marathon runner, right? This one's a sprinter, really fast off the block, but is a sprinter going to win a marathon against a marathoner? No, right? So it's going to be really fast to burn out. So for instance, just because I can lift up a really heavy textbook does not mean that I can hold it for long periods of time. That was a punishment when I was in school. They would make you hold textbooks straight up. This is Texas. They don't do things the way that you people do. If you kill somebody in Texas, they will kill you back. Don't think they won't. And I'm not saying it's good. I'm saying I grew up that way. Fastest to cause a contraction, least ability to remain contracted. The other extreme is smooth muscle is never in any particular hurry to get going, but it can maintain a contraction for a long period of time. It doesn't use up its ATP really, really quickly, so it can last for a really long time. And then cardiac muscle is kind of intermediate with both of those. It's not your emergency response tissue, but it needs to get in line so that it can support your emergency response tissue, right? And it can maintain a contraction intermediate between the two of those. So this brings me to what I call the diarrhea race. And the diarrhea race is really between these two, yes? And this one's just sitting around laughing at them, okay? So what happens when you eat something that irritates your digestive tract so much that your digestive tract tries really hard to get rid of it, right? So have you guys eaten at Shannon Thai? Just right over here. Really good Thai restaurant, but spicy AF, man. Like, seriously spicy. Like, the first time I went there, they're like, don't get above a 2. And I'm like, what the hell? I'm from Texas. Right? I can get above a 2. I can't get above a 2. Because I got 4, and I almost had to buy new pants. Okay? So I went in, got 4, thought I was all special and stuff, and all of a sudden my digestive tract, my smooth muscle, is like squishing down. It's not like in a super hurry, but it's not stopping. It's just like I said, squish, damn it. And the only thing that was saving my pants was this little bitty skeletal muscle at the bottom, right? This little skeletal muscle sphincter. And so to predict the winner, I had to decide whether I was going to engage in a sprint or a marathon, right? And which one could I win and save my pants? Sprint, exactly. You got to get to the bathroom quickly because that skeletal muscle is not going to be able to maintain that contraction for very long, and you're going to have to buy new pants again. No, I'm just kidding. And then the smooth muscle, if it's going to happen, isn't it? I mean, you've been a human for long enough to know that it's going to happen, right? If diarrhea is going to happen, diarrhea is going to happen either in public, which is awful, or in the bathroom, which is still awful, but less awful. So basically, it just depends on how you pit them against one another. But that teeny tiny little skeletal muscle is not going to last for very long. Okay, so, and then we already did the diagrams. All right, so last tidbit to talk about as we leave this set of notes is just to try to apply this to organs, right? Because we've been talking about tissues. But what I want to see is, can you apply this conceptually to organs? What do I mean? What I mean is, if I ran into a hollow organ, do I know what I expect to find on the inner surface, what I expect to find on the outer surface, what I expect to find on the wall? Do I also know how to learn from what I see? So for instance, if I ran into a hollow organ, you guys tell me first, what general category of tissue am I going to find lining the lumen? Epithelial tissue. But then there's something that can be gleaned from that. Because if it is going to be a thick epithelial tissue, is that probably going to be an absorptive organ? No? A really, really thick stratified epithelial tissue? No. Right? Probably protective. If it's a thin epithelial tissue, probably more likely to do transport. What if it had cilia on the surface? What does that tell you about what it's doing? It's moving something across it, right? Goblet cells will tell you mucus secretion, right? So you can put some structure function together with that logic. Like I know, does it make sense to you that the esophagus has stratified squamous epithelium? Yeah. It's continuous with, of course, eventually, the small intestine, which doesn't have stratified squamous epithelium, right? It's got a thinner, simple tissue, right? Right? Because you can put structure function together and figure, okay, this one's really not doing any transport, but it needs to be protective because Doritos are aggressive, right? They are, right? And then this one, right, everything's pretty much liquid by the time it gets down there, right? So we don't really need a whole hell of a lot of protection. So we can learn from the tissue that we actually see. So deep to that, and we'll get to these terms in just a second. Deep to that, you're going to find a basement membrane, of course, yeah? And then deep to the basement membrane, you're going to find what general category of tissue? Connective. Now, the connective tissue quite often is going to be a reeler. But would it tell you anything about the organ if it was, instead of a reeler, it was dense irregular? What would that tell you about the organ?
The way that it strips.
Yeah, so like something is hard on that organ. So which one do you imagine would be more likely to have dense irregular? The small intestine or the stomach?
Small intestine.
Stomach is dealing with solids, yes? And you stuff massive amounts in there at one time, like on Thanksgiving, right? You're like, how many mashed potatoes can I put in there? Do you at Thanksgiving, do you guys do this? Like, if I eat really fast, I won't get full before I eat the proper amount of mashed potatoes, right? Because some other little bastard in your family's going to eat them anyway, right? You know, and you don't even like that cousin. Don't give him your mashed potatoes. The connective tissue can tell you something, and if we make our way out, what we are generally going to find is if it's a hollow organ and it's not the heart, what's going to be here? If it's a wiggly hollow organ, it's going to be muscle, it's going to be smooth muscle, right? How many layers tells you what kinds of things are moving. The stomach has 3 thick layers. The small intestine has 2, and they're thinner, fluids versus solids, right? Does it surprise you that blood vessels have 2 relatively thin layers, right? And why 2? Well, this one right here, if the tube goes like this, and there's a layer of smooth muscle that wraps this direction, the fibers go that direction, then when it squishes, it's going to narrow the lumen, right? Squish the lumen. But then the next layer runs this direction, and when it contracts, it's going to pull the stuff down. That's how you do peristalsis through the digestive tract. You squish it together, and then you pull it. Squish it, pull it. And actually, that's what you were looking at, even though I didn't point it out to you. In this picture, right here, this is smooth muscle, but this is a longitudinal layer, long, skinny, and this is a cross-section of the same muscle, only it's running this direction. That makes sense? That's the double-layered smooth muscle. I would never ask you to identify anything on this one, but conceptually, can you see that that's actually the same tissue, cut 2 different ways? Yeah? Okay? All right, so, and then let's make our way to the outside. As we get on the more superficial surface of the muscle, I'm going to run into what tissue again? It's got to be connective, right? And then on the outer surface, there are 2 possibilities. Quite often, it is going to be an epithelial tissue, but some organs are stuck to other organs. They're like woven together. If 2 organs are, instead of hanging out by themselves like a stomach, right, they're actually stuck to one another, then what tissue are you going to put on the outside that will make it stickable? It's just going to be connective tissue, right? Okay, so for example, the ureters leave the kidney, yes? And when they leave the kidney, they run along the back side of the body wall, and they're stuck to the back side of the body wall. And the thing that sticks them is that they have connective tissue on the outside. But then they let go, right, and they dive into the body cavity, and then they're coated with epithelium when they're hanging out by themselves, right? Does that make sense? So on the outer surface, depending on whether it's stuck to something or sitting there by itself, it's either going to have epithelial tissue or connective tissue on the outside, okay? So let's just introduce you to these terms. I don't test on these terms on this test. I'm just going to tell you them because we're going to run into them all semester long, and it'll make you way more comfortable, okay? These particular terms, the mucosa, submucosa, muscularis, all that kind of stuff. So when you run into, when you get into the digestive system and they like look at a cross-section of the esophagus or whatever the hell they're looking at, right? So a really fast way to say inner layer, next layer, muscle layer, outer layer is the inner layer is called the mucosa layer because quite often it makes mucus, yes, okay? And it is basically whatever kind of appropriate epithelial tissue plus its basement membrane. And that's just generally the mucosa layer, and that's right there, okay, mucosa. Now, you can look at it and say, oh, it's got goblet cells, that tells me that. Oh, it's got cilia. Oh, it's thin versus thick, whatever, okay? So it's always called mucosa, but that doesn't tell you the kind of tissue it is. You have to kind of look at what kind of tissue is it. And by the way, we call these mucous membranes if they're moist. Mucous membranes are all of the moist membranes that are at least potentially exposed to the outside of the body. So like your whole digestive tract is a mucous membrane, the urogenital tract and the respiratory tract all have mucous membranes on the surface because they produce mucus at least some point. They end up being really clinically important because mucous membranes, aren't those 3 membranes the primary places you get pathogenic exposures?
Yes.
Yeah. Right, they end up being really clinically important. Okay, so deep to the mucous membrane is what we call the submucosa. That's a connective tissue of some sort, right? Okay, deep to that is this layer. It's not labeled here, but it's called the Muscularis Layer. And I won't ask you about these on this test. You're just going to meet them for the first time. And then on the outside, 2 possibilities. Either a serosa, because that's how you form a serous membrane, yes? Or if it's not a serosa and it's actually connective tissue, it's called adventitia on the outside, okay? So any organ, take a cross-section, don't be specific, and you're going to say mucosa, submucosa, muscularis, and then something on the outside, serosa or adventitia. Now, the reason I'm teaching that now is because once you get to the digestive system, when you're doing the digestive lab, you'll be like, what the hell? What are these words? And now you will know. Everybody else in your lab will be like, I don't even know what this word means. I'm just going to memorize it. Okay? But I don't ask you about these. I do ask you sort of this general thing, like what general category of tissue you're going to find the inside of a hollow organ, right? Where you can find next, that kind of stuff. Okay, and then last tidbit on this set of notes is, okay, we did a whole hell of a lot about tissues, and now we are ready to move on to organs. But the simplest organs are things that you don't think of as organs, and they are called membranes. So the simplest organs are even 2 layers of tissue on top of one another, and they don't seem like organs. But isn't an organ just more than one tissue functioning together, right? You learned that before, right? So, believe it or not, if I look at my liver is an organ, but then the serous membrane on the surface of my liver is an organ. Because the serous membrane is a combination of areolar connective tissue plus simple squamous epithelium, and that's an organ. Yeah? So let's look at that one really quickly. So serous membranes, you guys have already met them, you know them. They line closed body cavities, the ventral body cavities. And we know that there's basically 4 sheets, right? The big old windy one called the peritoneum, right? 2 of these, pleura, and then 1 of those, okay? 4 sheets. And then what is it composed of? It's consistent. They're always composed of a superficial simple squamous epithelium with its friend, the basement membrane, and then a deeper, loose areolar connective tissue. So they're made of the same thing all the time, okay? I always ask about this on tests. Oh, by the way, when you're studying for the test, any test, people miss everything on the last page of their notes. Because they flip, and they're like, oh, I'm done. And then I ask them the simplest damn thing from the last page of their notes, and they're like, I haven't looked at that in 12 weeks, right? So look at the last page of your notes at least every once in a while, because there's easy stuff there I could ask. Okay, the next type of membrane we've just been talking about. A mucous membrane is the membrane that is lining those moist inner surfaces of your body open to the outside, right? And what kind of tissue do you find on the mucous membrane? Well, the superficial layer is always going to be an epithelial tissue, but it's going to be whatever epithelial tissue you need there, right? Could be columnar, that's really common, right? Could be pseudostratified, could be stratified at the 2 ends, right? So it's not like a memorization thing, right? Mucous membranes are going to have, like in the digestive tract, esophagus, moist stratified squamous epithelium, and changes, a bunch of different kinds as you go through. And then at the other end, I'm going to have moist stratified squamous epithelium again. Yeah? And then I'm going to change as I go through. So that's not a memorization thing. That's like the epithelial tissue will be the appropriate epithelial tissue for the function. And then you're going to have basement membrane. And then you're going to have loose areolar connective tissue under that. Right? Almost always. There's a couple of exceptions, but almost always. And like I said, those are important common routes for pathogens. And then the last one is the skin. The skin is an organ. It's an organ because it has a really dependable superficial layer of what kind of tissue? Be specific, though. Keratinized stratified squamous epithelium. And then the dermis below, not the hypodermis, that's actually not part of the skin, but the dermis below is made of a combination of tissues. What are they? Dense irregular connective tissue, reticular, sometimes areolars in there as well. But either way, it's layered tissue, so it ends up being, right? It ends up being an actual organ. So the skin is an actual organ. We usually would cover skin right now, but I'm going to cover skin later because you need more help with bone than you do with skin, okay? And I'll come back and pick up skin before you have your lab practical. But we're going to do bone now because you guys need my help with bone, generally. Okay, so we are done with that set of notes and we are ready to start on the skeletal system. Okay, so we got to talk about skeletal system, what we do in lecture and what we do in lab on the skeletal system. Okay, so there are 4 chapters in your textbook that are relevant to skeletal system. There's chapter 6. Chapter 6 is like the development and big framework of skeletal system, like how it's all put together, how it develops, all that kind of stuff. I do a lot of that in lecture. It helps you to understand the big picture of the skeletal system. Chapter 7 and 8 are axial skeleton and appendicular skeleton. You're going to use those mostly for lab. Because do you think it's a really, really good use of your time for me to project a picture of the humerus and point out all the bumps? No, because you can't really learn it that way anyway, right? You actually have to learn that in lab. You have to touch it, you have to see it, right? Okay, so those you will mostly use in lab. I'm not saying I won't refer to them in lecture, I will. I'm going to use them as examples when I'm teaching. I'm going to use them to reinforce things. I am not going to take 2 hours to go through the skull in an image when you can actually do it in lab, okay? And then chapter 9 is articulations, which is like joints, right? And we'll cover a little bit of that with the skeletal system, and then we'll cover more of it when we move the joints with the muscular system. Okay, so here's something you guys need to understand. When we get to skeletal system, and also it's true for muscular system, every single thing that you learn about the skeletal system in lab, you are going to be tested on in lecture. Okay? So I am going to ask you all of the bones and bone markings, or at least I can, right? But within reason, because I know that the superior orbital fissure is really freaking hard to find if you don't have a 3D skull in front of you. So that's a terrible question to ask on a 2D image. But if you cannot find the foramen magnum, you are not conscious, right? Does that make sense?
Yeah.
So all of that to reinforce. So your test that has skeletal system on it will be mostly stuff from notes, but there will be a sprinkling of stuff from the lab. Likewise with your quizzes. Like I'll show you what kind of questions I'll ask from lab on your first bone quiz, right? Does that make sense? Okay. So consider all bones on your lab white sheets or lab manual a standing lecture assignment. And we're going to do the same thing with muscle. Is that I will reinforce them, I will use them for examples as we're going through. Okay. So, but I will help you. I'll help you figure out how to master the stuff that you're doing in lab. Okay, so first let's just briefly talk about the functions of the skeleton. So the skeleton, of course, is supporting, because it's supporting connective tissue. It's supporting the body. Without it, you would look like a flesh-colored pancake, resisting gravity. It is, of course, in some places protective, right? It's cranium and the ribs and around the spinal cord. The skeleton has a separate functionality that's directly related to the cardiovascular system because the red bone marrow inside your bones actually produces all of your blood cells. And that process is called hemopoiesis, or sometimes they call it hematopoiesis. They're both correct. It's just that people aren't very consistent about which prefix. And then your skeleton is a savings account for important minerals and also for fat. So yellow marrow stores fat. The bone itself actually stores calcium and phosphorus. And what do I mean by storage? So let's just talk about calcium. Calcium is obviously important for skeletal structure, but calcium is used for some other things that are arguably more important than just skeletal structure, like cardiac muscle contraction, yeah? And like causing release of neurotransmitters, right? So what would happen, for instance, if you didn't have adequate calcium in your diet? You would take it out of your skeletal system, right? And you would lose bone mass, but you would, of course, keep these other systems functioning. And that will and does occur. It will occur in all of us. It tends to occur, especially when in women postmenopausally, you lose skeletal mass, or if you have someone who has a chronic illness that's keeping them from eating, they're going to start to lose their skeletal mass, or someone who's suffering from anorexia. They're going to not just be losing fat and muscle, they are also going to be losing their skeletal mass. Does that make sense? And we'll talk about how that occurs at the end of this set of notes. And then the last one is that the skeleton doesn't move your body, but it provides leverage for movement. And what do I mean by leverage? So if I want to get something out of the ground, right, and it's like kind of buried, half buried in the ground, and I got a rope, I can actually tie a rope to it, and then I can use the ground as leverage for pulling it out, okay? The same thing happens with your skeleton using leverage for body movement, because this motion right here is using this bone right here as leverage to move this bone, right? The same thing when you talk about your deltoid muscle. My deltoid muscle does this by using my clavicle and scapula as leverage to move my humerus, right? So the skeleton itself doesn't move you, but it provides the stationary leverage to move another bone, right? That's not all it does, but it does that. Okay, so before we get into lecture-related stuff, I want to take some time and give you guys some tips for studying bones in lab, how to do it more successfully, how to make sure that you are going to be able to demonstrate what you know on your lab practical. So there are a couple things that are really helpful. First off, in your lab manual, if you bought the lab manual, there are 2 pages, I think it's like 34 and 35 or something, that has a bunch of meanings of bone markings, okay? This is a smaller one. There's more actually in your lab manual. And I know that it feels like you have entirely too much to learn, but I promise you that investing a little time to know the difference between a condyle, a tubercle, and a trochanter will help you remember when you have to write on your lab practical. Is this the occipital condyle? Is this the occipital tubercle? Is this the occipital trochanter? Well, you need to know what the word means so that you can figure out which one it is. For instance, a condyle is a rounded structure like a knuckle, right? Usually for forming joints. So how you make that meaningful to you is that you say, oh, that's the occipital condyle. And then you go find a bunch more condyles until you start seeing the pattern in a condyle, right? And that helps you to tell it from, for instance, a tuberosity, which is a big, rough projection, not smooth and rounded, right? So go find all the tuberosities, right? And figure out how you can tell. Because you've probably heard this, but let's tell you if you haven't. Your lab practical is practical, right? You have to actually look at the bone and identify the thing, and look at the muscle and identify the thing. It's also fill in the blank with no word bank. Okay? So you have to get a bunch of dependable ways to pull up those words in your vocabulary. And making them meaningful is going to help you, right? Instead of just rote memorization. So you are going to have a sheet, and it's going to have a tag on a bone, and this is going to start next week, and they're going to say, name this feature. And you're going to have to figure out which one it is, right? Is it a foramen? Is it a fossa? Is it a canal? Or is it a fissure, right? It's the optic something. How do you tell the difference? Oh, a foramen is a hole that doesn't tend to have much depth to it. A canal is a tunnel, yeah? Fissure is a slit, okay? And a fossa is a depression, doesn't go all the way through. And so that will help you figure out which one goes with it. Does that make sense? So this seems like more work. It will actually reduce the amount of work if you will memorize these terms, if you'll go find them, okay? So first, learn the meanings of the bone names. And then the next recommendation is once you find a type of thing, go find more than one of them, right? If it's a styloid process, and there's another styloid process. If it's a foramen, go find another foramen, right? Go find a bunch of them. Okay, and then the next recommendation is no longer just a recommendation, it's actually a requirement, because I finally got all the labs doing the same thing, and that is that all features must be parented to the bone. So, for instance, this is the zygomatic process. It's the left zygomatic process, but you have to parent it to the bone that it goes to. So you don't get to say zygomatic process. Eventually, when you're more confident, you have to say zygomatic process of the bone that it belongs to. So it's the zygomatic process of the left temporal bone. Does that make sense? Okay. You have to figure out what holes belong to, which holes belong to which bones, right? And it seems really impossible at first because the skull is all articulated, but there's 2 ways to really help you with this. Next time you're in lab, whether it's an open lab or your next lab, by the way, just because they do appendicular skeleton next week does not mean you can't get out the skull. You totally can. You can reinforce what you're doing. So when you get into lab next, how many of you guys have already been to lab this week? So not all of you, but when you get into lab next week, or if you go in there today, to figure out which bone things belong to is initially kind of challenging. It gets better. There's 2 ways to help you with that, I think. First off, colored skulls. I have 2 colored skulls on each campus. The colored skulls have each individual bone that's colored differently. So if I look inside the skull and I see kind of a hard bone like the sphenoid bone, I can see what it looks like from this view. I can flip the skull over and see the same bone in green. I think it's green. From this view, I can see it from the lateral view, and it helps me to get the boundaries of the bone in my head, right? Which is really helpful to know like what it looks like, because after you've seen it all separately colored, it's easier to find on a skull that's just skull colored. So I think that's really helpful, okay? And then the other thing that's really helpful is we have these exploded skulls, meaning the skulls in which all of the bones disarticulate, yeah? Okay, those are called Beauchene skulls. They're really expensive. You can totally play with them, but if something isn't going back in or coming out, stop pulling and ask somebody, okay? Because they're $1,200 a piece. Okay, but after you do that, you can take out the sphenoid bone, which is hard to see, and you can look at all the boundaries of the sphenoid bone individually, right? And once you've done that, that will actually help you click with what you're seeing in the skull, right? Yeah, you can. You can. You have to put it back together, though, or you have to be able to find somebody who will help you put it back together. It's not super fragile, right? But it's also not fragile, right? So if you're playing with a model that comes apart and it's somebody else's model, if it resists, don't push anymore, right? Go ask somebody, right? Okay. So the Beauchene skulls are really helpful. In addition, I want to reinforce the things that I've already told you, but now you're going to start to care. There are bones available for you to study in this library. There are bones in the STEM Center on Redwood campus. There are bones in the Jordan Library. There are bones in the library on Redwood campus. Okay, you can check them out. You can't take them home, obviously, but you can study them when you're in the library. How about buying a $25 skull off of Amazon?
Oh, no.
No, they don't have all the features. That's why they're $25, right? They don't. Our cheapest skull is $250, right? And then the one that's a little nicer than that is around $600, and then that one is the Beauchene skull, the exploded skull, is $1,200. And then we have two real skulls as well. You can't take those anywhere. No, they're really, really hard to get. Go ahead, ask your question.
I just thought, I was looking for electronic 3D. Don't we have what that goes with our book? Electronic files that go with it for these in particular?
Yeah, if
Okay.
you bought the expensive version of the textbook that comes with the digital
I
content,
bought the ebook. I'm not sure.
it should come with your ebook.
Okay.
Usually all the digital content comes with an ebook.
Okay. Sorry.
And I think it's called Complete Anatomy that comes with that.
Okay.
And then I also posted, did I post a link for you guys to one?
Yeah.
To one that's really good that they use in lab. Those things don't help me. I'm just like, what the hell? I like need to hold it. I'm like, oh, that's just pretty pictures. I feel like I'm playing a video game. I'm wasting my damn time. But some people actually learn really well from those things. They just don't really work for me. Okay, so you have to parent the bone. And that is not first tier learning. That takes a little while, right, to get to it. So you find the holes, and then you find the bone, then eventually you can put the holes together with the bone. But don't be super hard on yourself. First time through, grasp what you can. Second time, grasp a little more. You'll be fine. And then, so the other pointers for learning the bone. So starting this week with a couple bones and next week with a lot of bones, you are going to have to be able to figure out lefts and rights in addition to all of the parts of the bones. And it's initially really challenging, but there's a way to sort of master it more quickly. And that is, if I give you any bone, this guy right here, which is what you probably know what that one is. What is that? That's the femur. Okay, so even if you didn't know, you'll know it next week. It'll be fine. So you have to be able to tell left or right femur. This week, the only ones I think that left and right is challenging on is the clavicle. No, you do the clavicle next week. Just ribs this week. And the ribs are a pain in the ass. You just have to hold them and get used to it for a little bit, and you'll be fine. But and so next week, you're going to have to do lefts and rights on lots of bones. And it initially seems super hard. But here's the way that I recommend to do it. You can do it any damn way that works for you. But I know this way is pretty dependable. And that is, whenever you find a bone that you have to side, which is left or right, you have to be able to, from your perspective, find a dependable either anterior or posterior landmark. It doesn't matter whether it's anterior or posterior, because if you memorize this is always anterior, you can't see it, you're looking at the backside, right? This is always anterior. This little bump right here called the greater trochanter is the one that I use. You can use any one you want. As a matter of fact, the femur is actually kind of easy, and a lot of the long bones are, because your long bones will actually arch anteriorly. So if I lay it down, it's like a rainbow. If I'm looking at the top side, it's like a smile if I'm looking at the bottom side. Okay, So whatever you want to whatever you want to use. So you need anterior, posterior landmark. You, of course, need to tell top from bottom. On this bone, it's super easy. Can we agree? Like your kid could probably tell that that one is the one that goes to the body, right? Okay. And then you need to be able to tell lateral from medial. Yes? So you need to be able to have a lateral landmark or a medial landmark. Again, this is a good example of a bone because most of us would know that big head, and that's what it's called, is medial because it goes to the body, right? So why the hell does this matter? Because what you need to be able to do eventually, before you get to your lab practical, is you need to be able to do it without touching the bone. Not initially, but eventually. So you have to be able to empathize with wherever the person was. You have to be able to figure out, okay, where were their feet? Where are their heads? Are they face up or face down, right? And this is where you see people doing this when they're like trying to figure out siding of bones. They're like, what the hell am I looking at? So if you don't see the front side, you're looking at the back side, right? Medial versus lateral, and that will help you to side the bone. Does that make sense? And of course, at first you switch it around all you want, but I do want to tell you that at least half of the lab instructors do not let you touch the bones during the lab practical. And why do they do that? They do that because if she has the bone first and she flips it around, he actually gets a different question than her. And it might be more or less challenging, right? Does that make sense? So once you get to the later layers of studying, if you guys went to study together, what you would want to do is get out a box of bones in the library, the STEM center or whatever, and you pick a bone out and you put it on the table and you say, identify these features and tell me left or right, and then you try to do it from when she put it on the table. Now, when you put things on the table for yourself, you tend to do it the most comfortable way every time, right? You're like, this is the way I know this bone, this is the way it's going to be. But nobody has any idea how you memorize that bone, right? And some of them are kind of complicated, like the hip bones, the osteoclasts, and the scapula. They're kind of complicated. So go to the library, the open lab, or STEM center, practice pulling the bones out and identifying them without touching them. Position them differently every time once you get really used to it. And then try to figure out the position of the person, right? And then don't forget that you have a study sheet that nobody can take away from you for the bones, because there are lots of landmarks that you can feel. Yes?
Oh.
So, yes, you can feel. I can feel the styloid process of the radius right here. I can feel the lateral malleolus of the fibula. I can feel the medial malleolus here. I can feel the zygomatic arch, zygomatic process of the temporal bone, the temporal process of the zygomatic bone, especially for those of you who are kinesthetic learners, right? It seems dumb, and you might look dumb, but who the hell cares if you made an A, right? Literally nobody cares, right? So when you do that, it actually gives you another form of recall. The more senses you can involve in difficult study material, the better your recall. So in addition, you can use your own cheat sheet for a while to get to know things. One of my students who's, anybody really good at art in here? Drawing? Oh yeah, Charlie's an artist. So one of my students last semester I thought was fantastic. When she was studying, these little bastard bones right here are called the carpal bones, and they're really hard. So when she was studying, what she did was she drew the distal ends of the radius and the ulna, and then she drew the ventral view of the carpal bones and drew all of them in, right here, on herself with a Sharpie. And then she did the dorsal view on this arm, right? And she used them when she was studying until she really, really got it. Of course, she couldn't take that into the test, but she didn't try. So she just came into class with it and I was like, that's amazing. I got some pictures of it. It was awesome. So, so if you have a willing, enthusiastic partner at home and they're high five and a hell yes, not a meh, then maybe you can draw on them. And it'll turn into something more fun later. I don't know. You do you, man. And then the last thing is that you can't write it until you have written it. When you start feeling confident, and these two are studying together, and you put out a bone, and she correctly identifies the medial malleolus of the tibia, she needs to start writing medial malleolus of the tibia, because that is another learning technique. You remember the process of having written it. That's, again, kinesthetic learning. And sometimes, interestingly, have you ever had this experience when you're trying to remember something that you studied and you can't quite remember it until you close your eyes and you remember like where it was on the page or what color it was? So don't avoid the writing as a study tool and start doing it whenever you think I've got most of this down. Don't wait until the night before because you don't dependably spell things really well if you haven't spelled them before. And you need to be kind of critical with yourself about spelling. Spelling does count in lab. It doesn't count enough to drop you a letter grade, but it makes it a little harder to make an A if you haven't mastered spelling. Okay? Okay, so those are my pointers. Okay, and now we are going to change gears a little bit and talk about stuff that they haven't been talking about in lab or won't talk about. Go ahead.
Is this recording?
I think I'm recording it.
I was just
Yeah, it is right there.
Oh,
It just went away.
my side.
Yeah, just goes away sometimes. Okay, so the structure of bones. Oh, Jocelyn's not here today. I just realized that.
I only see her
You are
here,
not
so-
her babysitter, are
No,
you?
I'm not.
It's not
I
your
only see her.
Jocelyn, where are you? We noticed you're gone. Okay. Okay. They're brother and sister. They're not married. Okay, so what I want to talk about now is what bone is made of. And bone is basically made of, not surprisingly since it's a connective tissue, cells, fibers, right? And then the ground substance, but the ground substance in bone is specific. It's like the calcium crystals, yes? Okay. But what's interesting about bone is that bone is mostly not organic. Okay, so the organic component of the bone is the cells, meaning osteoblasts, osteocytes, osteoclasts, and then one more that you haven't learned yet. So that's organic, right? Organic is either living or made from living things. The collagen fibers that are in bone are also organic because they're made from living things. It's a protein, yes? But collectively, the cells and the collagen fibers only make up 35% of your bone mass. The rest of your bone mass is inorganic, primarily made of mineral crystals, mineral salts, calcium combinations, right, that come from your diet. And the minerals are inorganic because you don't need to eat living things to get them. If you licked a rock, you could get them as long as it was the right rock, right, into your bloodstream. Okay. So the organic component of bone starts with the cells. So let's look at the cells that make up bone. And 3 of these you met before, and there's one more. Okay, so the cells that make up bone, go ahead.
So does that mean that 65% is what gets depleted when we're taking
Exactly.
calcium?
Yes,
Okay,
uh-huh.
and the 35% doesn't?
Yes, the 35% doesn't break down very readily. And generally speaking, as long as you're not 85 or 90, you have the capacity to like replace it relatively readily because you can always make more collagen fibers, right? The only situation in which like that doesn't occur is supervillain have, what's it called? Unbreakable? Seen Unbreakable? Osteogenesis, no, what the hell, people? Osteogenesis imperfecta, which is a genetic disorder in which you don't form adequate levels of collagen fibers in your bones. And a lot of times when these babies are born, their clavicles or scapulas will break going out the birth canal, and they have tons and tons of breaks as they go through. Samuel L. Jackson. You don't see everything that he does. I love to hear him say mother effer. It makes me happy. I would say it in any other situation, but it makes me happy. I'm like, I gotta come up. By the time I'm 85, I'm gonna say that all the time. You can get away with it. So 4 kinds of cells. Sorry. These 3 are all part of the same cell lineage. Okay. We have these. These are called Osteoprogenitor cells. What is my progenitor? What is a progenitor? Yeah, a precursor, an ancestor. So my progenitors are my ancestors, yes. So this is an osteoprogenitor cell. It came from mesenchyme, yes. Then it differentiated enough that now it can only make bone stuff, including marrow cells, and then these, right? So this is the precursor to what we call an osteoblast. An osteoblast is the cell that is going to make matrix around itself. So we talked about this already. When it's young and not completely surrounded by matrix, what it's going to do is a couple of things. It's going to make collagen fibers and spit them out, yes. Then it's going to take all the calcium combinations from your bloodstream and it's going to dump them on top. Once it's completely surrounded and can chill a bit more, then it is an osteocyte and leaves a place for it to sit, which is a lacuna, right? Osteocytes can still help with bone matrix, but it's nowhere near as active in making bone matrix as an osteoblast. Does that make sense? So it's completely surrounded. It's an osteocyte. Okay. So those are the 3 cells that are involved in sort of making bone. And then this beastly looking thing right here comes from a different cell lineage, still from mesenchyme, by the way. But it is actually closely related to a macrophage. Do you guys remember macrophages? Those big old cell eaters, right? Macrophages are a type of white blood cell, which is also derived from bone, right? Okay. And what you do is to make this one, I won't ask you about this, but you fuse a bunch of them together, and then they make this big old beastly looking octopus thing, right? And what that one does is acts like a macrophage that's specific to bone matrix. So this is an osteoclast, and what it will do is it will break down bone matrix. For instance, if I didn't have enough calcium in my diet, but I still needed it for my heart, what these would do is break down the bone matrix, but where would they put the calcium? Back in the bloodstream, right? So they break down the bone matrix. So they're used for remodeling, right? They're also used when you need to make a withdrawal from your calcium bank or your phosphorus bank. Is that okay? So these are called osteoclasts, okay? And you don't see all of them unless you're specifically looking at a certain region of bone, but we will talk about seeing all of them. Okay, so, and then the collagen fibers, by the way, so here's an osteocyte right here. The collagen fibers would be around, right? It would have secreted it around. And then what about the stuff that is not collagen fibers and not cells? That is mostly calcium crystals. And the types of calcium crystals are 65% of the bone mass and primarily made of calcium phosphate, calcium hydroxide, calcium carbonate. And those deposit and they crystallize around. Calcium supplements are not at all uncommon. You've probably seen them in the grocery store or the pharmacy or wherever. There's several different kinds of calcium supplements that you can buy. So you can buy them over the counter, and some of them are like $10, or you can see one that's like $40. So what's the difference? The difference is that certain of these calcium compounds are very easily absorbed, and certain of them are not. So while it seems like the $10 calcium supplements are probably a good deal, you're probably going to pee out about 85% of the calcium because of the absorption mechanism. Okay, and you can look up which ones are easily absorbable really easily, but it has directly to do with how much they cost. Okay, and then the last thing that we will do today is just look at a long bone and the big picture structure of a long bone. And you guys are going to do this in lab next week. So we're going to learn all of this. So what do I mean by long bone? So we're going to look at the general structure of a long bone. And of course, this long bone is what? What is this one? The femur. And a femur is the longest bone in the body, but that's not what makes it a long bone. So let's talk about first what is a long bone. A long bone is a bone that is longer than it is wide, of course, but it also has to be round in cross-section. Okay? So obviously the femur, round in cross-section, longer than it is wide, right? But in addition, these guys right here, right? These are metacarpals, right? These are long, right? So they're longer than they are wide and long in cross-section, and round in cross-section. They don't have to be a certain length. They have to be kind of the certain proportions. And so obviously most of the long bones you're going to find in your limbs, right? Arms and legs. How about the ribs? Are those long bones? No. What are they in cross-section? They're flat in cross-section. Those are actually considered flat bones. Okay? So long bones. We're just going to look at a typical long bone. So when we're looking at a typical long bone, the first things that you will see, of course, and you're going to start doing this next time in lab, is that there's bumpy ends and then there's a long skinny middle. Okay? The bumpy ends are called epiphyses. Epi meaning upon. Okay? And you have a top one and a bottom one. Why are they not called superior and inferior epiphyses? Because they're on the limb. So what are they called? Proximal and distal epiphyses. Obviously, the first thing that you would need to do before you could tell the features of a long bone is to figure out which one's which. Luckily, with this bone, it's easy as hell, right? It is not with all of them, okay? The freaking fibula right here, that's a little bastard. It's hard to tell the top from the bottom on the fibula. But with this one, it's really easy. So proximal epiphysis and distal epiphysis. The epiphyses will give you fits because, of course, they are going to have a whole bunch of depressions and a whole bunch of bumps on them. Yes? Because that is where you attach muscles ligaments, tendons, form joints, and all of the little bumps are going to have names. Okay? So those tend to be a little challenging for students. They get a little better, especially when you know that what a tubercle versus a tuberosity versus a condyle versus an epicondyle looks like. Okay? And then the thing that goes through the middle is called the diaphysis. So epi means on or upon, dia means through. So on the ends through the middle. Diaphysis, also called the shaft. Does that make sense? Okay? Diaphysis or shaft. Okay, so the epiphyses and the diaphysis. So the epiphysis, by the way, the epiphyses are generally compact bone around the edges, spongy bone in the middle, and usually there's red bone marrow in there. Okay, not always, but usually there's red bone marrow in there. Okay, the diaphysis is generally compact bone, pretty thick all the way through the shaft. You always have a little bit of spongy bone right next to the cavity, but really thick, compact bone all the way down. Okay, that's what that's generally made of. And then in the middle is what we call the medullary or marrow cavity. And what's in here is, in an adult, yellow bone marrow that lightens the weight, because that would be really heavy if it was solid, right? So it lightens the weight. It's also fat storage, although you don't take the fat out of there unless you're freaking starving, okay? Like really starving, literally starving. Okay, so the epiphysis, and then we have the epiphyseal line. So let me introduce you to the epiphyseal line. So do you see this interruption in sort of the texture of the spongy bone right there? That interruption? Okay, what that is a remnant of where your growth plate, also called epiphyseal plate, used to be. Okay, so when you are still growing in height, length in your long bones, what happens is there is a cartilaginous plate between the diaphysis and the epiphysis, and the two cartilaginous plates on the end as you're growing, like if there's one at the distal end and there's one at the proximal end, as you grow they're going to move away from one another because you're going to continue to make bone mass. Okay, and then what will happen, and we'll see details about this later, is after you hit puberty, your sex hormones will start cause them to thin and thin and thin, right? And then eventually what will happen is they will seal. After they seal, you won't grow in height anymore, and you are going to have the remnant of the growth plate called an epiphyseal line. When it was open, it was an epiphyseal plate, okay? Growth plate. The epiphyseal line. Now, this ends up being kind of forensically important, right? Because if you find skeletal remains, whether they're human or animal, this helps you to age the skeleton. Because, of course, one of the first things you look for is it an adult or is it subadult, right? If it's a subadult, then at least in some of the bones, what will happen is as soon as it dries out, the epiphyses will fall off, right? Because the cartilage dries up, falls off. And a lot of times they get carried away by animals who won't have them. So you'll know that this person was under age 22-ish, right? A lot of times women stop growing 18, 19, men stop growing around 22. Okay, so they start a little later, right? Men hit puberty a little later than girls. And you can also do that if you ever find like the skeletal remains of a mammal, any other mammal, not human, right? We haven't found human skeletal remains. This ain't Stand By Me. But in the woods, you can figure out whether it's an adult or not an adult by looking at these. So that's called the epiphyseal line when it's closed. It's not always super easy to see. But a lot of times if you know exactly where to look for it, you can see it and you can section the bone. Okay, so then we have the diaphysis. That's compact bone surrounding a medullary cavity. And then on the inside of the medullary cavity, next week you guys are going to do like a really, really simple dissection. It's super easy. All we do is we go to the slaughterhouse and we get cow bones, long bones of cows, so that you can sort of see this stuff when it's fresh, instead of when it's actually all dried. So our bones are all dried. So what's on the inside, well, of course, you'll be able to see the difference between the super duper dense red marrow and the kind of squishy, fatty yellow marrow. And then you can dig out the yellow marrow and you can see that there's actually a membrane lining the cavity called the endosteum, endo, right? And the endosteum on the inside allows you to remodel that medullary cavity if you need to, because the endosteum is primarily composed of osteoblast, osteoclast, and osteoprogenitor cells, which means you can build it and you can take away from it, right? And that endosteum, by the way, doesn't just line this cavity, it also lines all of these tiny cavities, which means you can really get at the spongy bone if you ever need to, okay? So that's called the endosteum. thin membrane lining the medullary cavity and all other internal bones spaces. Okay, and then the last thing that we'll do today is these two, and then I'll let you guys go because you're a little ahead of the other class. Okay, so the periosteum is going to be the layer on the outside. So around the outside of the bone, do you see this? There is a layer of tissue that you are going to attach ligaments and tendons to, right? Does that make sense? And it's called the periosteum, which means around the bone. And you already know a little bit about it, don't you? Do you already know at least what one layer of the periosteum is made of, this outer layer that you're going to yank on? What kind of tissue is that made of? What is the periosteum made of? Dense irregular connective tissue, yeah? So the outer layer of the periosteum is made of dense irregular connective tissue because you can yank on the femur from a bunch of different directions. There's muscles attached on it on all different sides. But then you're going to have another layer that we'll talk about in just a second, and this layer is going to snuggle up to the bone. You don't yank on that layer. It's just against the bone. So the periosteum is a tough sheath on the outside of the bone. When you have a fresh bone next week, you can actually pull it off too. You can actually pull it off and see. And it's going to have a whole bunch of blood flow through under the periosteum right next to the bone. And it's going to have the ability to remodel the bone because the inner layer, called the deep layer or the cellular layer, snuggled right up against the bone, that layer is made of osteoblast, osteoclast, and osteoprogenitor cells. So now do you see that we can remodel the bone from the inside or we can remodel the bone from the outside, right? Okay. And the superficial or fibrous layer is, as you guys just told me, dense irregular connective tissue that I can yank on, right? From the outside. And it's an anchoring point for tendons and ligaments. It's a good tissue to put on the outside of something. And then, although we don't see it in this figure, there are a lot of blood vessels that sort of run under there to feed the bone. Okay, so we will talk about that next time. Okay, and then the last little tidbit for today is just what is at the end of the bone. At the end of the bone is going to be what we call articular cartilage. And articular cartilage is not telling you the type of tissue, it's telling you what it's doing. What does articular tell you that it's doing? Yeah, it's forming a joint, right? So that's why it's articular cartilage. Of course, in the vast majority of places, articular cartilage is made of what kind of cartilage tissue? Yeah, it's usually hyaline, okay? So the articular cartilage is usually hyaline cartilage, and hopefully we'll get some bones next week that still have some articular cartilage on it so that you can kind of see the texture of it. And the articular cartilage is really going to cushion so that two bones don't rub against one another, because we will see in this set of notes we talk about arthritis and how bones rubbing against one another will actually cause more bone growth and further restrict movement. Okay, so we'll stop there for today because you guys are a little ahead, and I will see you guys on Tuesday. And as soon as I get around to it, after your one quiz closes, I'll post another one so you can practice more histology.